So Parkinson's disease is a major problem. About 2% of us will develop Parkinson's disease at some point in our life. There are a number of small molecule therapies which have been developed to mitigate the symptoms of this disease. However, these small molecule therapies do not slow the progression of the disease. Toward this end, there are a number of clinical gene therapy trials which have been started using various viruses like adeno-associated virus packaged with neurotrophic factors like GDNF and others, which have gone through these trials. And these viruses can be injected into the striatum or the substantia nigra. While these trials have shown safety for gene therapy methods like these, they have shown limited efficacy. And it's been suggested that this limited efficacy is due to incomplete delivery to the targeted structures. We therefore hypothesize that we can improve the efficacy of these strategies by improving the delivery. So to improve the delivery to these structures, we must first cross two major barriers. The first being the blood-brain barrier, which we can transiently disrupt using MR-guided focused ultrasound. So down in the bottom here, in the bottom left, we show two representative MR images. Where on the left, we have a T2 image where we can clearly delineate various structures within a healthy rat brain. On the right, we have a post-focused ultrasound T1 contrast image where we can confirm the transient disruption of the blood-brain barrier. However, once therapeutics do cross this blood-brain barrier, they must then traverse an adhesive, nanoporous, and electrostatically charged extracellular matrix. Our collaborators at Johns Hopkins University have demonstrated that we can take nanoparticles as large as 114 nanometers, coat them in extremely dense coats of polyethylene glycol, and these nanoparticles can diffuse rapidly through this brain space. In the current study, we used a polyethylenamine, which is a cationic charged polymer, conjugated to a polyethylene glycol to condense a GDNF-bearing plasmid into about a 55 nanometer nanoparticle with a near neutral zeta potential. We tested this in a 6-OHDA rat model of Parkinson's disease, and this works by injecting 6-OHDA, which is a neurotoxin, directly into the striatum of the rat brain. This leads to about an 80% dopaminergic loss after about 2 weeks, which we can assess with behavioral tests. Finally, we will inject, or 2 weeks after injection of 6-OHDA, we will inject systemically our GDNF-bearing nanoparticles and treat the striatum using focused ultrasound. So in the T2 image on the left, you can clearly delineate the striatum, which is indicated in yellow. And then we use 4 overlapping spots of focused ultrasound to paint this entire striatal region. So 2 weeks after 6-OHDA, this is pre-focused ultrasound, we have less than 1 nanogram per milligram of GDNF protein content. 4 weeks later, at week 6, so this is 4 weeks post-focused ultrasound, we see a significant enhancement of GDNF protein content in our ipsilateral focused ultrasound treated brains. And these are the brains, excuse me, and the brains treated with focused ultrasound and GDNF nanoparticles. However, the animals that are treated with the nanoparticles only, or focused ultrasound and a non-therapeutic nanoparticle, or no treatment at all, remain at about 1 nanogram per milligram GDNF protein content. Importantly, we also do not see a GDNF protein content increase on the contralateral side. Furthermore, we see GDNF protein content remains high through week 12 after our treatment. Next, we can also do a series of behavioral tests. So on the left here is an apomorphine-induced contralateral rotation test. So the way this works, we inject apomorphine and the animal will turn in circles. In this particular model, a greater number of contralateral rotations per minute indicates a greater degree of Parkinsonism. So we treat the animals 2 weeks after administration of 6-OHDA, and we find that the animals that are treated with both our 6-OHDA, excuse me, our GDNF nanoparticles and focused ultrasound have a reduction in contralateral rotations per minute, indicating an improvement in locomotor function in those particular animals. However, control animals do not see a similar restoration. We can also do a 4-paw use bias test, where again, our only animals treated with focused ultrasound and our GDNF nanoparticles see a restoration in locomotor function on that contralateral side. We can also do serial sectioning of these animals in quantitative morphology. So on the left here, we can section the striatum. We can stain these sections with tyrosine hydroxylase, which is a marker for dopaminergic neurons. And we find that the animals that are treated with focused ultrasound and our GDNF nanoparticles have an increase in tyrosine hydroxylase staining density, indicating a greater density of dopaminergic neurons within these structures. Furthermore, in our substantia nigra pars compacta, we can also use similar staining methods to count how many cell bodies there are within that particular region. And again, we find that animals that are treated with our GDNF nanoparticles and our focused ultrasound have an increased density, about a 2 to 3-fold increase in density of dopaminergic neurons within these particular structures, whereas control animals do not have similar improvements. I just want to thank all our collaborators from UVA and Johns Hopkins and our grant support, including the Focused Ultrasound Foundation. Thanks very much.
Thank you very much.